We are going to look at the second stage of photosynthesis, which is the light-independent stage. Now, the light-independent stage occurs in the stroma. So if the chloroplast looks something like this, the thylakoid membrane is stacked up to form a structure known as the grana, and this is where the light-dependent stage occurs, and the rest of this space is known as the stroma. Now, the stroma is a gel matrix full of enzymes. So if we're to look at the equation again, these 12 H2O molecules were already used up in the light-dependent stage, and they were used to create two things, NADPH and ATP. Remember, ATP is the usable form of energy, and NADPH is a loaded acceptor molecule and is loaded with 1 hydrogen ion and 2 electrons. Now, we've already created the 6 oxygen gas molecules, which actually were released quite early in the light-dependent stage. So the main purpose of the light-independent stage is to create this really important product, which is glucose. So far in photosynthesis, this 6CO2 have been waiting to be used, and they're going to make an appearance now in the light-independent stage. So here are our inputs needed for the light-independent stage: 6 carbon dioxide molecules, NADPH and ATP. Now, the process that occurs in the light-independent stage is known as the Kelvin-Benson cycle, named after the scientist who discovered it. And the ATP made in the light-dependent stage is used to power this process. The first input that we see into the Kelvin-Benson cycle is carbon dioxide. Now, you can see that there are 6 carbon dioxides occurring here. I'm only going to talk about one at a time, but you know that 6 are coming in, therefore this occurs 6 times. So carbon dioxide enters the Kelvin-Benson cycle and fixes in a process known as carbon fixation to a 5-carbon molecule known as ribulose biphosphate. Ribulose biphosphate circles around in the carbon-Benson cycle. So here you can see the 5-carbon molecule, which has 2 phosphates attached, and you know that it's going to have 2 phosphates because of the name biphosphate. Now, carbon dioxide needs help in order to fix to this 5-carbon molecule, and the help comes in the form of rubisco, which is an enzyme. Rubisco is the most abundant enzyme on Earth. So, rubisco is used to help bind carbon dioxide, which looks like this, a carbon molecule bound to 2 oxygens, to this 5-carbon molecule, ribulose biphosphate. And what actually is formed then is a 6-carbon molecule. So this 6-carbon molecule can be seen here, and remember, it does still have the 2 oxygens which have come from this carbon dioxide, and it has the 2 phosphates that have come from this ribulose biphosphate. Now, this 6-carbon molecule is unstable, which means it isn't happy being in this formation. So what actually happens is it splits up into 2 3-carbon molecules which are stable. And these 3-carbon molecules are known as phosphoglycerate, or PGA. Now, you notice here that these 3-carbon molecules still have an oxygen and a phosphate as well. Now, when we're talking about making a carbohydrate, which is the purpose of this stage, is to make glucose, we know that we need 3 elements, and those elements are carbon, hydrogen and oxygen. So we've got carbon here, we've got oxygen. The one element that we're missing is hydrogen. So that's when the NADPH from the light-dependent stage makes its appearance. Now, this NADPH, which we obtain from the light-dependent stage, donates the hydrogen that it's carrying. And by doing that, this actually becomes NADP again, which is an unloaded acceptor molecule, which shuttles itself back to the light-dependent stage to take another hydrogen to then come back to this stage, so it's being recycled. So when this hydrogen ion joins this phosphoglycerate, it makes a new molecule, a 3-carbon sugar phosphate molecule called phosphoglyceraldehyde, or PGAL. Now, this molecule can actually exit the Kelvin-Benson cycle here, where it joins with another PGAL molecule, which then means that it's got 6 carbons, and we know that a 6 carbon goes towards making glucose, to create a sugar molecule called hexose, which is used to create glucose. Now, some of these PGAL molecules actually stay in the Kelvin-Benson cycle, and they are used to regenerate ribulose biphosphate that stays in the cycle. So that's how ribulose biphosphate is able to continue in that cycle, because it is continually being regenerated by some PGAL molecules that don't leave. And so that is the light independent phase of photosynthesis.